Hello everyone and welcome to today's webinar, Stability Testing as Quality Control Measure, Optimizing the Process Throughout the Product Lifecycle. I'm Tim Wright, Editor of Contract Pharma, and today's presentation is being sponsored by Frontage Laboratories, a CRO providing integrated, scientifically driven product development services throughout the drug discovery and development process, enabling biopharma companies to achieve their drug development goals. Our speaker today is Dr. Gary Dong. He's the Executive Director of Analytical Services of CMC at Frontage. And in this role, Dr. Dong is responsible for overall operations, scientific achievement, and regulatory rigor of analytical support of drug product development services to clients. He oversees analytical method development and validation, stability studies, and routine testing in support of clients' IND, NDA, and ANDA needs. He has over 20 years of industry experience in the analytical area under both R&D and QC environments. This webinar today will look at why assessing a compound stability is a complex and lengthy process, with objectives varying by the development phase. Dr. Dong will discuss specifically the purpose, scope, and type of stability testing required at each phase of product development. With this understanding, sponsors can aim to optimize the process and ensure that the right data are gathered at the right time. We will be accepting questions throughout the presentation. You can send those in using the box at the lower left of your screen. Questions will be answered after the presentation in brief Q&A, and this presentation will be archived on our website, contractpharma.com, for 1 year. And if for any reason you're experiencing technical difficulties, you can type those into the box as well, and someone from the broadcast vendor will help you out. And if you're ready, Gary, at this point, I'll turn it over to you.
Thank you so much, Tim, for the nice introduction. Good morning, everyone. Thank you so much for attending this webinar. I'm actually going to skip my bio and move directly to the outline of today's webinar. So I'm going to spend some time talking about stability in drug product development cycle, and also talk about stability guidelines, and also analytical methods validation and stress study, photo stability study, EU stability study, in stability support, and also going to spend some time talking about drug product specification set up for stability study, and stability design at different stages of drug product development. And also I'm going to talk about bracketing approach, matrixing approach, and data evaluation. Okay, this slide shows the flow chart of drug product development. Stability study is an important and integral part of the drug product development cycle. For preclinical stage, normally we conduct excipients compatibility study to understand the general stability nature of the API with the excipients, and we understand which excipients we should hang on with and which we have to stay away for our future formulations. So the early phase stability studies normally at IND phase 1 and phase 2, and we focus on collecting quick and basic stability information of the drug product. The data collected can provide directions for future product development and manufacturer process control for better and stable product. At this stage, we also do stability support to prove the clinical trial material are good to go with the study. During the early IND phases, stability study failure may lead to reformation and redevelopment of the drug product. So stability properly designed with R&D samples at extreme conditions such as high temperature and even including light stress conditions can provide quick and useful information regarding the drug formulation stability for the screening purpose. The late phase IND stage, in IND phase 3 and phase 4, the stability study are designed to collect drug product stability information to understand the best storage condition of the drug product and help us with packaging configuration, design and selection, and to provide the support for drug product shelf life estimation. The NDA registration normally occurs at the late phase stage of NDA phase 3. The stability study at this stage are fully regulated per ISH guideline to ensure that only safe, pure, and effective drug products will be approved and marketed. After product approval, the stability study is a continuous commitment to monitor the market drug product stability. Stability studies are required for post-approval changes such as excipient vendor change, manufacturing site change, to demonstrate that the changes will not impact the product stability. Okay, this slide shows the ISH idea first started in 1989 and became official in 1990 with the initiative to harmonize regulatory requirements and guidance for pharmaceutical product development and submissions requirements. European Union, USA, and Japan are the three founding members, and later on, Canada and Switzerland joined ISH as standing regulatory members. Currently, there are many countries and regions and also organizations join the ISH as regulatory members and standing observers. Stability requirements are also written into the government laws. For example, here, like Q1A actually is the most important document, which actually deals with the stability study design, storage condition, and sample testing intervals and submission requirements. And other ISH guideline documents, as their titles indicated, address specific areas in the stability study and provide regulatory guidance to the industry. And Q1F here I just want to mention, discuss about stability data package and the registration applications in climate zones 3 and 4. Somehow this guidance document was withdrawn by ISH because countries in East Asian and African, they actually want to apply different storage conditions which will fit their climate conditions better. So right now, ISH cross-references the WHO requirements in this area. Okay, so stability studies are also written in the government law. So here is the detailed requirement from Code of Regulation or Federal Regulation from the USA government. So I just read the requirement. There shall be a written testing program designed to assess the stability characteristics of drug products. The written program shall be followed and shall include sample size, the testing intervals, and storage conditions of the samples, and testing procedures involved, and also the testing of the drug product in the same packaging configuration as your marketed product. And if there is any requirement, we also have to perform in-use stability study testing. In this slide, I'm going to show the general content of a stability protocol, followed by Frontage Lab and also the industry in general. So the first one is the cover page. Normally we have protocol identification, protocol name, and approvals. And in the introduction section, we describe briefly about drug product. In the stability setup section, so we list detailed information about batch selections and also the packaging configurations of the drug product. And in the stability schedule, we have tabulated information about sampling schedules and storage conditions. And we also list the detailed information about testing procedures and acceptance criteria. And this is a GMP document. For sure, we have deviations reference documented, and we also document the revision history of the stability protocol. So this slide shows the definition of the climate zone 1, 2, 3, and 4. So The majority of the countries from Northern Hemisphere are covered by Zone 1 and 2. For example, in North America, USA, Canada, and Mexico, and also countries from northern part of Asia and Africa also belong to this group. Many countries from Southeast Asia, Turkic area, Africa, and South Hemisphere are in Zone 3 and 4. So where in general the climate condition is hot, dry, or hot, humid. So for different climate areas, there are different stability conditions requirements from the SH guideline. So this slide shows the general storage conditions requirement for climate zone 1 and 2. We follow this storage condition. So here I just want to point out for the long-term storage condition, so there is an alternative storage condition at 30 degrees and with 65% relative humidity. So if we do apply this as the long-term storage condition, so there is no requirement for intermediate storage condition. If we do keep our samples under refrigeration, and the accelerated condition will be 25 degrees, 60% RH, and there is no requirement for intermediate storage condition. And for drug products, we must keep under freezing condition. The long-term storage condition only is minus 20 degrees. And this slide shows the storage condition requirement for climate zone 1 and 2 for products in semi-permeable containers, and most apply to aqueous-based products or non-aqueous-based, solvent-based products. And here I want to point out the storage condition for the humidity requirement. So at long term, it's 40 degrees, 40% RH, and alternative storage condition at 30 degrees and 35% relative humidity. And for the accelerated condition, the humidity requirement is no more than 25%. So here I do have a question to everyone. Please pay attention to the humidity requirement for intermediate storage condition. So is that requirement right? So this slide shows the storage condition requirement for climate zone 3 and 4. And for the long-term storage condition, there are two alternative storage conditions. Both are at 30 degrees, and the first one with 65% relative humidity, and the second one with 75% relative humidity. So the condition you pick up depends where you want to market your product. The rest is the same as the climate zone 1 and 2. For the stability testing frequency for the samples, the guideline is straightforward, very simple. The long-term storage condition for the first year, we test at every 3 months. So we have initial 3 months, 6 months, 9 months, and 12 months. And once we move on to the second year, we test at every 6 months. So we will have 18 months and 24 months. And afterwards, it's annually. So we will have testing points at 36, 48 months, and so on. For the accelerated condition, the ISH guideline requirement is a minimum of 3 time points. And for a time period of 6 months, the suggested testing time point is initial 3 and 6 months. We recommend to do a little bit more time points. We can do initial 1 month, 2 months, 3 months, and 6 months. And more data points from accelerated condition will help you estimate better the long-term storage condition for estimation of the drug shelf life. For the intermediate storage condition samples, so we will only initiate the test once we observe significant change at accelerated condition. So the ISH guideline requirement is a minimum of up to six months testing. So we suggest, keep testing intermediate storage sample as long as it's required. So this will provide better information for you to analyze the stability data to give a better estimation of the drug product shelf life. So all the analytical procedures used for stability support must be validated. So here is a list of the tests. So we'll have assay, the impurity, by HPLC, chemical purity. We also have preservative antioxidant by HPLC for semi-solids, and performance tests such as dissolution for solid dose, and drug release for oral solutions and oral suspensions. And other tests, for example, physical test of viscosity for your semi-solids. And this is a summary of all method validation recommendations. Analytical methods validation depends on the development stage of the drug product. As early stage R&D phase 1, the drug product formulation is very simple. The developed analytical procedures may not be continuously used for R&D phase 2. So shall we do validation or not? So what is the FDA requirement here? So FDA mentioned in their guidance document for R&D phase 1 requirements that laboratory tests should be scientifically sound, specific, sensitive, and accurate, and they should be suitable and reliable for their specified purpose. So if you do talk the requirement with FDA auditors, they will politely answer and use a magic word, validatable, for this requirement. So in general, we recommend conduct validation, but we do want to stay focused on the basic performance of the method itself. So we generally can skip away from intermediate precision study and robustness study. As we move on to R&D phase 2, the drug product formulation is more complicated. The formulation and the dosage forms may remain similar for future. FDA requirements in this stage is appropriate validation data should be provided. At this stage, we do recommend conducting a little bit more validation on top of the R&D phase 1 package. We suggest to add on the intermediate precision study also. Once we move to R&D phase 3, the drug product development is a mature stage. So the formulation is firmed up, and the manufacturing process has been systematically studied and decided. And at this stage, we recommend full ISH guideline validation, including everything. The force degradation study is recommended for all stage of the drug product development for stability support. So the purpose for force degradation study is to prove your analytical procedures, they are stability indicating, and also we collect information to understand the impurity profile and the degradation pathways. And for R&D phase 1, we recommend to do the force degradation study, but some clients may want to stay away from force degradation study at this stage. So we recommend, if you want to stay away from force degradation study, do prepare yourself with the drug product and the procedure force degradation study data to back up yourself. For R&D phase 2, it is highly recommended to conduct force degradation study. And once we move on to R&D phase 3, so the force degradation study is mandatory. We must conduct force degradation study. This is a list of typical force degradation study conditions we follow here at Frontage Lab, and the industry follow the same in general. The thermal stress, we normally use elevated temperatures, 50 to 60 degrees, and sometimes even higher. And for acid and base hydrolysis, we apply hydrogen chloride and sodium hydroxide for up to 24 hours, and normally end up in the solution concentration of 0.1 normal to 1 normal. And for oxidation, the condition is straightforward with 3% hydrogen peroxide for up to 24 hours. So here, just remind, do stay away from high temperature once you do the oxidation. And for the light stress, ISH guideline is followed, but in general, we can use stronger conditions up to 3 times of the ISH guideline suggestion. And when we perform force degradation studies, so we have kept in mind to reach reasonable degradation around 10%. 10% is also in line with the policy specification requirement in general. And normally, if we actually can achieve somewhere from 5 to 20% is ideal. But absolutely, we shall never overstress the drug product to be more than 30% degradation. Mass balance is also required by regulatory agents during the submission. If mass balance is significantly off, you had first analyze the potential reason for this one. So if you can support yourself scientifically, for example, the off balance at higher end is caused by one of the diagrams with a very strong irrecoverable metal, so you are fine. But if you cannot explain this one well, and you should redesign the stress condition and reconduct force degradation study. Photostability study is also required specifically and also rating the ISH guideline. So the purpose for photostability study is to evaluate the intrinsic photostability nature of the drug product itself, and also to demonstrate the photolabile drug product is sufficiently protected from light. And all drug products, particularly for semi-solids, oral solutions, and suspensions, we had conduct photostability study. So it is also required after we make changes in the formulation and packaging configuration of the drug product. The light source requirement is per ISH guideline. So for the samples to test, we recommend design and include all the following. So the directly exposed sample, so that's the naked drug product in quartz containers. Primary pack, that's your sample in primary packaging configuration. Marketing pack, that's the samples in primary and the secondary packaging configuration. And the dark control, that's the naked drug product in quartz container wrapped up with aluminum foil. And when we design the study, we also have to pay attention to the design factors. And we want to minimize drug product physical characteristics change, such as evaporation and phase separation, melting. And for solid dose drug product, so we have to evenly spread in a single layer. And we also have to make sure drug products are uniformly exposed to light. For example, if we do have tubes, so we have to make sure we use different orientations, horizontal and vertical, for the study. And all the tests from the drug product specification, except MLT and the sterility test, should be conducted for photostability testing. EU stability. EU stability study, the purpose is to establish a period of time during which multiple drug products can be used while retaining quantity within an acceptable specification range once the drug product container is opened. So there is no black and white guidance from ISH. EMA has a note for guidance on the subject. It suggests to conduct of the in-use stability study with 2 primary batches when you do the submission. And in-use stability study is required for all stages for ND study and for NDA and ANDA submissions. And we recommend, and this also required, that at least we have to do the in-use stability study at the end of the stability study program or at the drug product shelf life. When we have multiple strengths of the drug product or the drug product, they are packaged with different packaging configurations, we have used the worst case scenario approach. So we test the drug product, which is the easiest one to show degradation. So for in-use stability study design, we normally stimulate patient use. We had to conduct dilution, reconstitution according to the label requirements. And we also have to pay attention about the daily removal or withdrawal of the sample, and this should be included in the protocol. And the sample handling and the storage condition normally is conducted at uncontrolled room temperature. So a lab environment is ideal for this. And the test interval depends on how long we design the in-use stability study. For example, if the stability is designed for 1 month, we can do full testing intervals, including initial 1 week, 2 weeks, and 30 days for the study. All the tests, from the drug product specification should be included, for in-use stability study, including MLT sterility test. And if we do observe any issues with the stability for the in-use study, so we must include the information clearly in the labeling information. For example, we can label one drug product as keep under refrigeration after dilution and use in 7 days. Drug product specification is the same as method validation, evolves and depends on drug product development stages. So what is the FDA requirement for R&D phase 1? So in its 1995 guidance document, it is clearly stated no need for specification at the initial stage of drug product development. At this early stage, for most of the specification limits, we can simply use report for information. However, for assay, we can use a limit of 90.0% to 110.0% for clinical trials intended to conduct in USA. If you want to do the clinical trials in European countries, potency specification of 95.0 to 105.0 label claim should be set up. For R&D phase 2, we can differentiate, known and unknown impurities and follow SH guideline Q3B for impurity control limit. Other tests, depending on the obtained stability results from quick R&D trials, we may propose some numerical range limits, but they should always be set up at the loose end to allow further evaluation from the stability study to fine-tune the specification limits. For R&D phase 3, the product specification should be more firmed up to provide basis for the registration batch requirements. Specifications should also reflect the intrinsic nature of the API. For example, if the API is proved to be unstable, so its degradation products shall be well studied, including necessary toxicology study, so proper limits of the known degradants can be set up. They might be even higher than the SH guideline recommendation, depending on the nature of the toxicology of the degradant itself. Product formulated for topical applications may have more generous limits regarding the impurity control. The solution specifications will be different for immediate release, controlled release by nature, and they will have very different requirements. Our final specification limits should be supported by collected stability data of the drug product and meaningful. The purpose of the limits is to ensure that we have a safe, efficient, and pure drug product for the patient use. ISH guideline Q3B is a very useful guidance to set up impurity specification limits for NDA products. As discussed, if a known impurity is carefully studied and evaluated for its toxicological nature, its limits can be set up according to the supportive findings. For NDA product, simply we just follow the market product specification. So if the market product is not available, so we can use the general concept for NDA. So for the stability study for R&D stages, for R&D study, so we can use extreme conditions such as higher temperature at 50 degrees and use open bottle and sealed bottle, and accelerated storage condition, light stress condition if necessary. Set up study for a time period of up to 2 months, and we can do T0, 2 weeks, 4 weeks, and 8 weeks. 8 weeks. This purpose is to collect quick information about the stability nature of the drug product itself. And for phase 2 and phase 3, and we can apply the same concept, use the extreme condition. We can also include long-term conditions for R&D samples. And if we do the stability study for the GMP demo batches or for clinical trial materials for phase 1, we suggest to set up samples at accelerated condition and long-term condition. Long-term condition, we normally set up at testing point of the initial 3 months and 6 months. This will cover the clinical trial study requirements. But we do want to keep the samples and test as necessary if the clinical trial will last longer. For the accelerated condition, normally just do up to 3 months to test at 1 month, 2 months, and 3 months. Stability study for registration, the purpose is to understand the retest and expiration date of the drug product itself, and also find out the long-term storage condition, and also what is the impact of environmental conditions such as heat, humidity on the drug product itself. ISH guidelines are very clear about batch selections. We need at least 2 pilot batch and 1 smaller batch, totally 3 batches for NDA submission. So the formulations and packaging configurations and manufacturing process should be similar and representative of the commercial product. And the batches we select for the stability study should represent each individual strength and each individual packaging configuration. And for stability consideration, we follow the ISH guideline. And for semi-solids, solutions, and suspensions, we normally keep the long-term samples to at least 24 months with the option to extend if it's necessary. For solid drug products, we keep the long-term testing to at least 36 months with the option to extend if it's necessary. And at this stage, we suggest carefully check if in-use stability study, freeze-thaw study, leachable and extractable, and IVRP tests are required. For stability study for ANDA, for R&D stage, simply follow the requirement for R&D submission, and if necessary, include extreme conditions. And here I just want to point, do include the RLD samples in the study. So this will provide very useful information if you do observe unexpected impurities. For the GMP study for the submission, use the same consideration for the NDA submission. Bracketing and the matrixing approach are widely used for stability study design to cut the sample requirement and testing requirements short. And bracketing approach, normally we use extreme design factors, and we assume the intermediate samples are represented by the extremes. And for the matching approach, it's a little bit more complicated. We actually have selected the subsets, tested, and specific testing points. And we assume subsets represent the stability of all the samples. And at the time of submission, we do have all subsets tested, and we also test all subsets samples at the end of the stability study. And the requirement for this approach would be the formulation and packaging configurations are the same or similar for all samples. Bracketing and matching approach, they are very useful too for drug product development, particularly for formulation with multiple strengths and with multiple packaging configurations intended to different markets global-wise, and we can save a lot of budget and also use the data information for product stability evaluation. However, if we do the filing for NDA and ANDA submission, should we use this approach? Based on our experience, we suggest do not use either bracketing or matching approach when you file for your submission for NDA or ANDA. Always include all samples of different strengths and packaging configurations for all the required test intervals in your stability design. However, if you do want to use the approach, carefully discuss this one with FDA in your pre-submission meeting. Only use this approach when you get positive confirmation. So for data evaluation of the stability results, and there is a SH guideline from SHQ1E, and 3 points I want to bring the attention to everyone. The first one is data variation and significant change. So we have to pay attention regarding this one, and also statistical evaluation. So the trending analysis with the static approach, if we do observe the trending line with 95% confidence level with the acceptance criteria of the drug product, so this will define the drug product shelf life. Pooled data is also allowed if analysis show that batch-to-batch variation is small. And extrapolation applies well with the data is consistent with later variation and cannot be proposed if significant changes is observed at accelerated condition and at intermediate condition. So this is a decision tree from the SH guideline. So if we do observe, the first question we check is if we do have significant change at accelerated condition within 6 months. If our answer is no, that means our product is stable. And the next one is we check the long-term data. So if the data show little or no change over time, or little or no variability at all. So if our answer is yes to both, that means our product is very stable. And the next we have to check is the higher temperature accelerated data. So our question is the same, little or no change over time, and little or no variability. If our answer is yes to both, that means our product is extremely stable. So in this scenario, even the statistical requirement analysis is normally not required. So we can claim 2 times of the shelf life claimed by in the long-term storage condition. And if we do observe a little bit change, variation of our long-term condition, so next question we ask ourselves is if the long-term data amenable for statistical treatment, or is the static analysis performed? If we do not have this, so we can only claim a 1/2 time shelf life from the long-term storage coverage. And if we do perform statistic analysis and support ourselves, and we actually can claim longer product shelf life. So this is going to be 2 times the coverage from the long-term study. OS and OT and OE results. And first of all, stability study is designed to collect solid evidence to prove that our product is stable for the given period. So by nature, we do expect OS, OT, or OE results if our samples are stored long enough. If we do observe OT results, first question we have to ask is we follow our company requirement for the investigation to check if they are confirmed or not, if they are caused by identified causes such as instrument or human errors. So if they are confirmed OS results, the first question to ourselves is if they are truly product related. Some might be simply, method related, particularly for the early drug product development stage. The drug product methods will not be thoroughly studied. For example, maybe for the different degradation products, they may co-elute or elute closely to each other. The impurity will be calculated as one unknown and cause the OS results. So if the OS results are product related, so the formulation and analytical scientists should openly and effectively communicate to each other and work together to nail down the cause. So this might be formulation related or process parameter control related. So this gives us the opportunity to further improve the formulation and the process control. And sometimes, the results may simply caused by the nature of the API. The API is not stable and sensitive to light, for example, or moisture, which can lead to speedy degradation. Then consideration of the drug product packaging configuration should be considered to help us to improve the drug product stability. OT, OE observation do give us the opportunity to investigate the potential problems or hidden issues at the early stage during the drug product development. So we can also set up alarming stability specification limit for our stability study. This will enable us to investigate this abnormality sooner than later. So we should put efforts in systematic OT, OE investigation in the early development stage to avoid development of drug product with questionable stability. And we should use OT, OE investigation as a tool, an opportunity to guide us to develop a more stable drug product. So to conclude, stability support is critical during product development cycle, and we must design product-specific stability studies to catch and discover potential stability issues at early stage. Analytic method validation and requirements and stability specifications should be set up properly and updated to fit the needs of different phases of drug product development. We also talked about the general requirements for photostability study and EU stability study, and regulation requirements and guidelines must be followed. Our goal is to deliver safe, pure, and effective drug product. So this is a list of the references for this webinar. And I want to thank everyone for your time attending this webinar. I also want to take this opportunity to express my special thanks to Dr. Kang Wang, VP, Analytical Services, Dr. Dongmei Wang, Executive Vice President, Global CMC Services at Frontage, for the discussion and suggestions to format this webinar. Thank you again, everyone.
All right, great. Thank you so much, Gary. And yeah, so everybody, we have some time here for some questions, so we'll jump right in, get to as many as we can. The first question, Gary, is there any need to conduct accelerated stability study for longer than 6 months?
Our answer is no. It's usually no. So there is no need to conduct accelerated stability studies after 6 months. Accelerated condition data is used in general to evaluate drug product stability and help estimate the drug product shelf life. For the testing at accelerated conditions after 6 months is not required by SH guideline. And also, according to the Arrhenius equation, and we know at higher temperature, at 40 degrees, the average degradation reaction rate is almost 9 times faster than at 25 degrees. So the 6 months accelerated stability data already gave us more than enough requirement, for the long-term shelf life estimation.
Okay, thanks. Moving on. Next question asks, should we do method validation for analytical procedures applied for IND-1 stability support?
For this question, the answer is definitely yes. FDA guideline for IND-1 requires all the tests, should be scientifically sound. So the only proof for us to meet the FDA requirement is through the method validation. However, we do recommend conduct only selected basic method performance parameter studies, as we discussed in the webinar. So you do not need to do full SH guideline validation at this stage. And in general, as I mentioned during the webinar, we can stay away from the intermediate precision study and also the robustness study.
Okay. And let's see, what's next here? What is the difference between CFR and ICH guideline requirements for stability study?
This is a very, very good question. So the basic difference is the first one is the government required law requirement. And if you do not comply with the requirements, so there will be very severe punishment. For the SH guideline, there are recommendations and relatively specific, for the practice to fill the legal requirements. And there is no direct consequences, but there are indirect, consequences, and they might be also very, very severe for non-compliance.
Okay, thank you. And how do you conduct force degradation study for a combination product with 2 actives?
Okay, this is a very complicated situation, and it's a very good question. And in this case, the force degradation study is very, very difficult, and particularly if you have drug products with 2 actives with very different, strengths. The way to conduct a force degradation study shall be specifically to the product itself, and we should study carefully the chemical nature of the activities, and always treat the approach with a case-by-case scenario. And I would suggest the following steps to conduct a study, and carefully study the nature of the API and use the information to help yourself to design the stress conditions properly, and also clearly mark the known impurities as impurity 1, as the active 1 or active 2 related. So if you do have the information, for example, from the filing information of the drug substances. And when you design the study, so you can include APIs alone and include placebo with one API only as controls. And all these samples can help identify the origin of the detergents to see which actives they are originally coming from. And also calculate the mass balance separately for the two APIs. So this approach works fine for all impurities observed, if they can be correctly identified, as either from active 1 or active 2. And if they are not 100% assigned to either of the actives, you calculate the impurities, use the base of the lower strength API. And once you finish everything, and you tabulate your data, very carefully, and then do the mass balance everything. And good luck, when you try to explain everything, to an FDA auditor.
Okay, great. Let's see. Moving on here. Next question asks, how do you deal with OOS, OOT, OOE results from stability studies?
And in the last section of my webinar, so we discussed about how we evaluate OOS, OOT, and OOE results from the stability study. So the general requirement is straightforward, as explained, and we should follow our company's SOP to investigate the results to confirm if they are OOS, OE, or OOT. And questions we should ask ourselves is, are these results method related? And there is a chance, might be particularly for early phase studies for drug product development. And we also have to analyze the information to check if they are product related. And we can use this as an opportunity for us to improve the drug product formulation and also the process control parameter and the packaging configuration.
Okay.
And we also have, yeah.
I'm sorry, go ahead. I thought you had finished.
Yeah, it should be fun. Yeah, so we should use the OS investigation opportunity as a very useful tool, for the formulation and process control to improve the drug product.
Okay. Are you there, Gary?
Yep.
Okay, sorry, it was cutting out on my end a little bit. Sorry about that. Okay, so the next question is, if drug substance API shows very stable stability, can drug product stability for early phase study be omitted?
This one, the answer clearly is no, it cannot be omitted. FDA guideline for industry for phase 1 investigation states, we recommend initiation of stability studies using representative samples of phase 1 investigation drug to monitor the stability and quality of phase 1 investigation of drug during the clinical trial. So this is clearly required by FDA. We had to conduct the stability. And also for AMD phase 1 material, even with very simple drug product formulation, such as powder in bottles or powder in capsules. So the clinical trial material we use is not exactly the same as the pure API itself. So we do need to conduct our official stability study to prove that is the clinical trial material itself is stable during the clinical trial study under the real conditions and packaging conditions we applied for the clinical trial material.
Okay. Let's see, next here is, what's the definition of significant change for stability data?
A significant change during the stability study can be found from the ISH guideline. In summary, basically, if you do observe physical change of your drug product, and you observe, 5% or more change in the processing result of your results compared to the initial results, or if you have dissolution, you actually cannot pass at stage 2, or simply you have OS results in the stability study. So all these actually are defined as a significant change.
Okay, and is there any requirement of minimum number of batches for stability data evaluation?
Okay. This is a very good question. There is no official requirement for R&D stage, and in general, some of the early phase clinical trials, maybe one batch is good enough for clinical trial itself. So you do not have any option. You have more batches for stability data evaluation. But if you do have multiple data points for your stability study itself, you can still apply the statistical analysis of the data evaluation. However, for NDA submission stage, the stability data requirement must be conducted on all the batches used for the submission. So the minimum requirement is 3 batches. So all the real data, should be included for submission. So from this, the requirement, is minimum 3 batches.
Okay, thank you. And I think we have time for one more question here. And this is regarding a generic stability program. Do you still do force degradation if the data is available for known impurities? Are you there, Gary?
Yes, I'm here. Yeah. The answer is yes. Even the force degradation study is available. We still recommend include force degradation study for your program. And the excipients, everything you use, for the formulation might be different. Suppliers from different origins, and this may give hidden issues, for drug product stability.
Okay, great. All right, thank you so much, Gary. I really appreciate your time and expertise. That's all the time we have. So thank you again, Gary, and to our sponsor, Frontage Labs. And as I mentioned earlier, this session will be archived on our website, contractpharma.com, for 1 year. And if there were any questions that we didn't have time to get to, the folks at Frontage will follow up with you directly. And thank you very much for joining us, and have a great day, everybody. Bye-bye.
Okay, thank you.